It's my extreme pleasure to introduce Mo Khalil. Mo got his bachelor's at Stanford in mechanical engineering before moving to MIT, where I first met him, where we were both in Angela Belcher's lab at the time. After that, he moved to Jim Collins's lab at BU, which he stayed on as a faculty. And he's really done a tremendous amount of work in his career, really at the, looking at epigenetics and chromatin, but also really just like engineering in general as a tool, really across fields. And, so it's a lot of very interesting things, a lot of different areas. He's won a number of awards, the New Innovator Award, most recently a Schmidt Award, where he is now delving into the world of plants. So, like I said, it's going all places. And so with that, I'll let him take it away. And thanks again for coming to this speak.
Thank you, Galen. Can you all, thanks. Thanks, Galen, for that nice introduction. Can you all hear me in the back okay? Sorry. Okay, fantastic. Great, it's really nice to be here. First time to Madison, having a lovely time. Unfortunately, it's a short trip for me. I'm back on a flight, later tonight, but I've really enjoyed getting to know some new friends and colleagues, and to catch up with a lot of like older and very familiar faces. So thank you so much, Galen. Thanks for being a great host. Reginald, thank you for organizing, and thank you all for being here. What I'm going to do today is kind of tell you a little bit of our recent forays in the field of synthetic biology. I don't have psychedelic movies like Scott Coyle, but I hope my talk will stimulate just as much. So first, the most important slide, I get to work with a really fantastic group of scientists just every day. This is a little bit outdated, but this is a picture of the group right on the Charles River between Boston and Cambridge. And I'm going to return to this slide when I note the individuals working on the projects that I tell you about. So that group and myself are really inspired by the remarkable biological functions that are carried out by cells. And of course, one of the finest examples are cells of the immune system, like this T lymphocyte pictured here in this really nice movie by Eric Betzik's lab. And as you know very well, these cells survey diverse tissues. They recognize a whole set of pathological insults. They actuate really targeted responses and even mount broader responses like mobilizing the immune system. And over the years, of course, we've come to appreciate that these functions like sensing, computation, actuation, et cetera, are really regulated by these complex networks or circuits of interacting molecules in the cell. So as synthetic biologists, we're really excited about the potential of, in a way, reconstituting these complex functions. And it's wonderful to be here talking to a biochemistry community because, I don't have to tell you that the concept of reconstitution, of course, is not a radical or conceptually new idea. Researchers have been, of course, reconstituting biological processes for, in some cases, over 100 years, right? In the test tube, for example. What I think is a new concept, and the one that we're really excited about, is the idea of trying to actually reconstitute, in a way, quote unquote, reconstitute these behaviors actually inside living cells, right? Where we can kind of preserve the advantages of biochemical reconstitution, but now do it in a cellular environment where we can address some of the more interesting and complex, maybe not interesting, but more complex behaviors that cells do, okay? So that's kind of our mission. And so over the years, what we've done is tried to develop tools to do this, synthetic biology tools that allow us to construct, quote unquote, orthogonal regulatory circuits in living cells. What I like to joke is that we're trying to basically turn biochemistry into a genetics problem. And I think that this offers a lot of really interesting and transformative possibilities. And that'll be sort of the outline of my talk. The first possibility that I think is quite interesting is we can use this as a somewhat of a discovery tool. It provides a really interesting and unique way to understand how we get emergent behaviors right, from the coordinated activities of molecules, right? And trying to sort of learn something about the quote unquote principles or the design principles of these regulatory circuits. And I'll focus the first kind of third of my talk around this. And we have really focused on, applying this approach in the context of eukaryotic gene regulation in particular. Okay, so the first project I'm going to share with you was led by Megan Bragdon and Nikit Patel, and in collaboration with my long-term, colleague and friend Caleb Basher, Yane Kondev, and others. So let me just start by saying, right, gene regulatory circuits orchestrate the intricate gene expression patterns that determine cellular identity and function. This is what gives us the phenotypic variation, around us, right, between species and even within species. And an important layer of that gene regulation is transcription, orchestrated by transcription circuits that basically turn signals into the transcription of genes, right? Kicking off this process. And this is, of course, many researchers are interested in understanding the form, the function, and the evolution of these types of networks. At the most basic level, these kind of quote unquote circuits are formed by the interactions of regulatory proteins called transcription factors that bind to small pieces of DNA called cis-regulatory elements to, of course, drive transcription. But what we find really fascinating about this problem, this is actually a non-trivial problem, just even that, right? Because these interactions are occurring in the context of a complex eukaryotic genome where there are potentially many possibilities for off-target binding and regulation, right? And so that's what I say, this is a very non-trivial problem that can really impact the fidelity and impact the function of these circuits. And we know this is true because of the many disease-causing mutations that lead to altered transcription factor binding and transcription factor activity, such as, for example, mutations to c-Myc and getting elevated c-Myc in tumors and cancers, right? So this leads to this very basic question: how do cells establish specific circuit connections to maintain circuit function? All right, so what I'm going to do in one slide is give you kind of decades of work of what mechanistic and genome-wide studies have kind of taught us about some of the principles, right, of these circuits. Okay, first, eukaryotic transcription factors, unlike their bacterial counterparts, tend to bind short and highly degenerate sequences, okay? That are basically, not sufficient to encode specificity in the genome alone, right? They do this with highly conserved transcription factors and transcription factor binding sites, basically using the same quote unquote parts. In addition, transcription factors, distinct transcription factors tend to be co-bound or clustered upstream of promoters and enhancers. And finally, this animation was good when I put it together, but now it's a pain. Okay, here we go. Cooperative interactions and cooperative binding among those transcription factors tends to be the rule rather than the exception, okay? And I think, one good way of defining what I mean by that is, parents of twins will know we get energetic coupling, between these transcription factors that enhances the activity or the binding of one, okay? And in fact, in extreme cases, we see the cooperative assembly of complexes of transcription machinery through these sort of multivalent interactions between these components, such as pictured here in the classic enhantiosome. Okay, so these are really interesting ingredients. How and why does nature use them to build circuits? So I'd like to propose that actually synthetic biology and studying this question with synthetic circuits is an interesting way to investigate the problem. And the reason is because introducing a synthetic circuit or a synthetic network into a cell, for example, a mammalian cell or a yeast cell, is somewhat analogous to disrupting the system like these mutations that I was telling you about, right? And can destabilize the system and present opportunities for misregulation. And in fact, my colleagues in synthetic biology, a whole subfield has been kind of devoted to sort of understanding this crosstalk between synthetic and natural systems that can have a major impact on engineered function, right, of these living systems. So we've set out to investigate this with a platform that we've been developing since really we started the lab. And it looks a little bit like this, and I'm going to gloss over some details. We're working in the model, budding yeast. And in yeast, what we can do is engineer and express synthetic transcription factors built from these basically artificial zinc finger DNA binding domains. Okay, so we can build these inducible systems where you can add a chemical like estradiol and lead to the expression of these transcription factors that are fused to transcription activation domains that can recruit transcriptional machinery that can be recruited to a reporter locus to drive transcription. Okay. And so if you do experiments like this and activate the circuit like this, what you'll notice if you measure cell growth very carefully is, for example, circuit gets turned on, but you get a concomitant, in a way, impact on the fitness of the host organism. In other words, you get a fitness defect relative to sort of the background strain, right? It's growing. It's physiologically impacted and growing less well. And if you then do, for example, a transcriptomic analysis of these cells that are carrying this circuit where you express them, what you notice is, maybe not surprisingly, widespread kind of dysregulation, right, of the transcriptome that's associated with this fitness defect. Okay, so this is the phenotype that I was telling you about in the previous slide that provides, I think, a promising way to study this problem. Okay. So one can gain some, very simple intuition for this problem by building just a mathematical model, the simplest possible mathematical model for transcription regulation. This is one of these thermodynamic models where you can consider a transcription factor and a target site, right? As well as other, let's call them off-target sites. And then you can use equilibrium considerations to compute the binding and the transcription at these different loci, okay? And what's nice about these models is because they're biophysically kind of meaningful, you can then ask the question, on target versus off target regulation and plot that as a function of different biophysical parameters. In this case, the affinity of the transcription factor to DNA and the level of cooperativity between the transcription factors, okay? And I think it's shown very clearly here. What you see is basically an increase in that space of specificity on target versus off target as you lower the affinity of transcription to DNA, make them weaker, and increase the cooperativity, make them more dependent on one another, okay? So again, we're of course missing a lot in this model, but as a first principle, we gain some intuition. Great, so how do we test this experimentally now? Okay, so let's return to this platform and this model system that I introduced earlier on. And now I'm going to add a little more detail to it. Okay, so again, we have inducible expression over these 3-finger transcription factors, right, that are activating some reporter gene. We, over the years, have identified residues in the zinc finger protein, these arginines, that mediate nonspecific interactions with the DNA phosphate backbone. What's nice about these is you can now go and mutagenize these, in this case to alanines, and you can actually mutagenize these in a systematic way to tune the affinity of the transcription factor to DNA across a nice physiological range. And when you do that, right, and you just measure reporter, what you can see here is, of course, the lowering of reporter activity, which is associated with making these transcription factors basically weaker, right? So, and here's the second feature of this platform that we developed. To now control cooperative interactions between these transcription factors, we developed a scheme that's based on this synthetic protein we call the clamp, which is basically a small protein of covalently linked PDZ interaction domains that bind each of these PDZ domains will bind a C-terminal ligand that we affix to the transcription factor. Okay, so without getting into a lot of detail, what's nice about this system is that you have full control over the valency of the complex and the biochemical interactions that comprise it, okay? And so what I'm showing you here in this plot is reading out kind of the dose response of the reporter as we increase the size of the complex using a low affinity transcription factor from 1 to 4, right? And you get these characteristic hallmark cooperative shifts, and of course, a restoration of the transcriptional activity, okay? So hopefully I've convinced you that we can engineer cooperative factors in yeast. Okay, great. So now let's go back to that data that I showed you before. I'm just plotting it differently here. This is the data that I showed you before of these, high affinity we'll call them wild type transcription factors that led to the fitness defect. Okay, so as I showed you before, circuit is on, we get reporter activity, relative fitness is low. If we just introduce those mutations that lower the affinity, what you see, I think, really nicely is basically a restoration of that fitness back to sort of background levels. The cells are now happy, but of course we've disabled the circuit, right? These are weak transcription factors that are unable to stimulate transcription. And I think you're probably already anticipating the punchline, which is when we co-express the clamp to make them dependent on one another and now reinforce assembly at that locus, we restore transcriptional activity, putting ourselves in this nice, what I think of sort of as an optimal space, biochemical space of both high fitness and high transcription or high circuit activity. Here was the RNA-seq data that I showed you before. Now I have to do the experiment of showing you what that looks like, when we express, the low affinity transcription factors and the kind of the cooperative low affinity transcription factors. And what you can see here is, largely an erasure or a restoration of that misregulation of the transcriptome that's associated with the improvement in fitness. But to really complete the model, Megan did a heroic effort, which is to develop a ChIP-seq protocol for these synthetic transcription. Basically, try to chip down these transcription factor and measure binding in a way, quote unquote, occupancy or binding across the genome. And anyway, over a couple of years, she finally got a really nice protocol for doing this. And I'm showing you a lot of data here. What we're plotting are these, the high affinity, low fitness, right, circuits versus the low affinity and cooperative ones. And the picture, the kind of the general message here is that what we generally see is what you might expect, which is promiscuous binding to a bunch of different endogenous, sites by the high affinity transcription factors. That's largely, quote unquote, erased when you lower their affinity, making them weak, and then sharpening their activity right at the reporter. Okay. Great. Okay, so the next thing we became interested in studying, and as you can imagine, that fitness defect that I've been talking about could have an impact in quote unquote, real world applications of these circuits, right? For example, if one were to sort of grow long-term some of these yeast cells in a bioreactor, you can imagine you could, select out your engineered function, right? Because they have a fitness defect. Same with sort of expanding, let's say, an engineered cellular therapy product. And so to sort of investigate this a little further, we took these circuits and we cultured them in a continuous culture device that was developed by Brandon Wong in the lab a number of years ago. We call it Evolver. I don't have time to tell you a little bit tell you more about this system. But if you guys do any microbial continuous culture or continuous directed evolution, it's a really interesting platform and it helps to sort of solve some of the deficiencies in scale and flexibility of continuous culture. But here's kind of what some of this data looks like. So this is basically running over hundreds of hours of continuous growth, these yeast strains that I showed you before. And what you can see very immediately, I think, is that when you induce the circuit, an immediate loss right, of the population growth rate, right, in the kind of, I'll just call them the bad circuits for now, just to be colloquial, and then a restoration of that growth rate that would be consistent with the idea of the accumulation of mutations to the circuit that restore, fitness. And if you measure properties of the circuit like the transcription factor concentration or the circuit output, what you can see here is basically a gradual loss of the transcription factor and the emergence of this basically this disabled circuit population, consistent with what I've just described here. In contrast, the cooperative circuits are growing happily and on, for the duration of this 100-hour multi-generational experiment. So, we go a little bit further in the paper. You should check it out if you're interested, but we isolated, of course, some of these genotypes, map the mutations. It's very interesting. Basically, we get a set of mutations that really target the transcription factor, both its activity as well as its expression. Basically, the cell, I hate to say it this way, but it's like attacking the transcription factor, right? And if you take those mutations and install them back into the sort of low fitness circuit, one by one, I love this result. I mean, the transcriptome just goes right back to background levels, okay? So these are in fact driver mutations. All right, so let me kind of conclude this part of the talk. So what I showed you is using sort of synthetic biology. Studying this problem, we found that specificity in regulatory networks can emerge from cooperative interactions among proteins that individually interact weakly and non-specifically. This is a possible explanation for solving this kind of quote-unquote specificity paradox. How is it that eukaryotic transcription factors mediate such precise regulation in development, right? Despite recognizing short and degenerate motifs? And the answer is, you can do it when you collaborate, right? I showed you also that synthetic circuits can really have an impact on the cellular host, right? And this could be problematic for applications of synthetic biology. However, cooperative assembly seems to be a simple and generalizable strategy. You can install this strategy onto lots of different transcription factors or various other types of regulatory networks and enhance the genetic and functional stability. Remember, all we're doing, just weakening affinity and making them dependent on one another, okay? We're not doing exquisite protein engineering. In prior work, in a paper published a number of years ago, we also showed that cooperative assembly is a really flexible mechanism for engineering diverse and complex signal processing behaviors. Check that out if you're interested. But I think the best way to kind of sum up this work is this proverb, this famous proverb, right? That helps to explain why complexity like this may have evolved, right? If you want to go fast, go alone. If you want to go far, go together. These results give me kind of a way to answer a personal question. Why the hell did you have 4 kids? More is better. That's why. I always get a couple chuckles and people are like, oh my God, he has 4 kids. All right. I won't get into this because I won't dive deep here, but I put this slide together because just in the last year, there has been really amazing work in the community, in the literature, highlighting the importance of cooperative and low affinity binding among transcription factors and transcription regulation that I just wanted to highlight. This is not our work, this is others' work. But if you're interested, check it out. I'll just highlight this one. This is from Emma Farley's lab at UCSD. She studies these vertebrate enhancers, important in development, of course, and they contain just littered with low affinity transcription factor binding sites. And what she found was human variants in these enhancers that are associated with disease. Those mutations optimize, they actually increase the affinity of the transcription factors And when you install them in mice, they cause polydactyly, right? What it's saying is evolution has been suboptimal, right, in binding interaction. Okay, I think that's such a fascinating result. And of course, there's a lot of other work in this space that you should check out. At a higher level of organization, we know that transcription circuits and gene regulation, broadly speaking, depends on, of course, histone modifications and chromatin organization. Galen mentioned that our lab has had a long history of studying problems in chromatin using synthetic biology approaches. This classic, for example, the genome is organized in terms of domains that are heterochromatic, that are kind of off, transcriptionally silent, euchromatic, transcriptionally on, right? And Colin, in a nice collaboration with Kaushik Raghunathan's lab at Brandeis, has been taking these ideas of cooperativity and crosstalk and now applying them to study crosstalk between DNA modifications and histone modifications using synthetic approaches as well. Next time I come back, I'll tell you all about that story. But I get to put my twins up here again. All right. Great. Is it okay if I shift gears a little bit? All right, excellent. So the second part of this talk, I want to tell you about how the concepts of developing tools to reconstitute these functions can be quite useful for developing technologies and synthetic circuits to enable what I'll call just precise and instructive control of therapeutic cell function, okay? And here, really what we want to do is coax cells like immune cells, which already have disease-modifying potential, right? To make them more specific, to make them more controlled, and to make them better therapies. This has been a really fun, big-scale project. It was originally sort of pioneered by Huishan Li and Divya Israni. We've accumulated a number of amazing collaborators along the way: Wilson Wong, Cole Roybal, and Keith Jung. And I think I don't have to tell you all that we've entered a pretty exciting era in medicine. With the emergence of gene and cell therapies, maybe the poster child is the CAR T cell. This is the idea, of course, of taking patient-derived T cells, engineering them to express synthetic receptors known as chimeric antigen receptors that can then allow them to be redirected to desired tumor antigens to then unleash the anti-tumor immunity and anti-tumor potential of T cells. And many of you know that this modality has shown some remarkable activity and some results in particular B cell cancers, but of course has a lot of shortcomings and limitations, even in addressing solid tumors, with some of the challenges listed here and reviewed here. But of course, as just the leading edge for many, maybe some other diseases, there's going to be a lot of work needed to be able to make these safer and more effective therapies. And this has motivated synthetic biologists and many people, of course, to think about the idea of how we could maybe use things like synthetic gene circuits, right, to address some of these roadblocks that I was referring to before, and perhaps expand the therapeutic capabilities of these human immune cells. There's many reasons why one might want to impart control over these, right? These are kind of autonomous therapies that are injected into patients. And so, of course, if we can increase the control of specificity, this would be useful, right? Solid tumors, unlike B cells, right, don't have these like lineage-specific defining antigens. So it becomes harder to figure out what the discriminatory antigens are for solid tumors. And of course, it would be nice to be able to sort of really modulate the efficacy of these so we don't get into situations where you get cytokine release storm or overactivity of the immune system as a result of these potent products, et cetera, et cetera. Okay. So my lab and others have been excited about the idea of trying to realize this vision. And the way we think this will be possible is by developing synthetic biology toolkits that we're going to call, quote unquote, clinically optimized, right? And what I mean by that is trying to develop tools that fulfill some sort of basic criteria for potential clinical applications. For example, maximize the use of humanized components and proteins when possible to reduce the risk of immunogenicity, engineer constructs that are minimized in size so they can be delivered with standard delivery techniques, et cetera. So this kind of framework for us really, and these design criteria converged on a basic building block for engineering these gene circuits. And that's at the very, at the core level, these zinc finger domains or these zinc finger proteins. So I've referred to them earlier in my talk, but I'm going to provide a little more background now. Galen and I joke that everyone's forgotten about zinc fingers because of CRISPR, but I want to remind you, right? So zinc fingers are DNA binding domains. They're in fact the most prevalent DNA binding domain in all human transcription factors. They're extremely prevalent. And this suggests that, of course, this is nature's solution to the problem of DNA binding and DNA recognition, and that because they're humanized, they're less, have less of a potential risk for immunogenicity, for example. Zinc fingers can be designed to recognize new sequences and concatenated to bind larger sequences. And this dates back to work from even the early 90s, especially following the solving of the crystal structure of ZIF268 by Carl Paabo. Of course, not as simple as just making a guide RNA, but it can be done. In addition, zinc fingers have actually been, as a precedent, been shown to lead to long-term viability in kind of long-term regulated expression of genes in non-human primates and other preclinical studies. Okay, so this is why we like zinc fingers for this application. So with Keith Jung's lab, a preeminent zinc finger engineering and genome editing lab at Harvard, we set out to design a set of zinc finger proteins that recognize pieces of DNA that would be as distant as possible, quote unquote, distant as possible from human genome sequence, making them quote unquote orthogonal. So this is just showing you, these 11 motifs that we designed where, for example, there's no occurrence of an exact site in the human genome sequence, okay? And with Keith, we set out to design basically 6-finger proteins that recognize these motifs, which when fused to human transcription effectors like P65, form the basis of what we call these synzifters or synthetic zinc finger transcription regulators. Okay, does all make sense? Okay. So I'm kind of glossing over a lot of characterization of that protein engineering. You'll have to take my word for it or look at the paper. But I want to just show you how we proved the platform. And the idea was to use these synzifters to demonstrate that they could be used to engineer control of therapeutic T-cell activity, using CAR-T as our kind of paradigm here, okay? And what I mean by user control is in effect trying to realize this vision of, for example, allowing a user or a physician to modulate the activity of the T-cell using, for example, a small molecule or something that could deliver to the patient, all right? And there's many reasons why this may be useful. Being able to sort of pause T-cell activity has been shown to be really powerful by Crystal Mackel's lab and many others in Wendell Lim's lab to sort of limit T-cell exhaustion and make these more potent. And I'm just kind of outlining a lot of the virtues of this type of solution that's been, again, shown and reviewed elsewhere. So what we came up with is this way in which we could develop synzifters that could be regulated by small molecule drugs. And in particular, we prioritized small molecules that are already FDA approved. I think you can imagine why that might be. You can use them in sort of an off-label way and repurpose them now. We know them to be safe. We know them to have good pharmacokinetics, and then use them to regulate the activity of these synthetic zinc finger transcription factors. Does that make sense? So here's two flavors. One is regulated by the FDA-approved antiviral protease inhibitor called grazoprevir, a really interesting drug. It's taken at really high dose by patients. So we can make, for example, a synzifter switch that gets activated in the presence of that small molecule. Here's another switch that we developed that's regulated by tamoxifen, the well-known breast cancer drug, or metabolites of tamoxifen like 4-OHT. Okay. So does this even work? Okay. So we used these systems to now develop switches or circuits in primary human T cells, such that, for example, in this case, using grazoprevir to control the expression of a chimeric antigen receptor. In this case, the model HER2 CAR. Okay, so some data in primary cells. So this is just in vitro data. This is showing you really nice expression of the CAR on T cells in the presence of the drug to levels that are close to, for example, constitutively expressing this. This is in vitro killing data. So when you co-incubate these T cells with leukemia cell lines expressing your antigen, you get drug-dependent killing of those tumor cells, of those cancer cell lines. And then in vivo, in mice, establishing a xenograft blood tumor, and then dosing those mice with these engineered T cells. What I'm showing you here is effective tumor control and actually tumor eradication when we dose the animals with grazoprevir versus no drug, showing that we have full drug-dependent control over the activity of these T cells here. I think what's nice about this system is it's not limited to just controlling CARs. You can control other payloads. So here's an example of building a switch where we use, let's say, the other drug, tamoxifen, to control the activity of a proliferative cytokine, IL-2. And actually, by the way, there's many reasons why you might want strong sort of dose control over cytokines like IL-12 and IL-2, which can be quite toxic at high doses. So same sort of data that I showed you before. Here's tamoxifen-regulated IL-2 production in these primary T cells. This is showing you if we dose for different amounts of time, we can increase the proliferation of those T cells in vitro, right? And then in a really nice in vivo model that Huishan developed, she would dose these animals with these engineered T cells and then give them tamoxifen and then go count, basically, T cells in vivo at different compartments. So this is showing you we have control over the expansion or the proliferation of those engineered T cells in the blood and the spleen, for example. Again, showing this kind of on-demand control over proliferation. So this is, I think, a really fun, fun kind of chapter in this work, because this now provides ingredients to build more complex types of circuitry in primary and really therapeutically relevant cells. And here's just one kind of fun example. We imagined a situation where in T cells that were engineered with a double switch, we could, for example, add tamoxifen to proliferate those cells, expand those cells, add a second drug to turn on anti-tumor activity. Does that make sense? That wasn't good. Here we go. Sorry, I was getting too excited. This is just flow cytometry data showing we do, in fact, in individual cells have dual control over these different payloads. And then Huishan developed a complicated but fascinating in vivo model to actually test this control circuit, which we're calling a sequential control circuit. And I can't get into a lot of details here because I don't have enough time. But the basic idea is that she would inject into the animals. This is my boundary. I'll remember it. Okay. Inject into these animals first the T cells, okay, and then wait for a duration of time, and then challenge with the tumor. What this does is it increases the challenge, the difficulty of trying to control that tumor, because the T cells have been just basically waiting around with no antigen stimulation. Does that make sense? And so that's a great way to test for this sequential control circuit, because now if you do a sequential dose, you expand them using the drug, and then you turn on, right? The hope is that you would get anti-tumor activity. And in fact, that's what I'm showing you here, at least in these survival curves. We can prolong the survival of mice when we dose with this sequential dose of tamoxifen, then grazoprevir. Again, a very artificial model. There's nothing like, I think, clinically relevant about that, but it's supposed to sort of captivate the imagination of what types of control circuits one could build in now primary T cells using this platform. All right, so let me sum up this portion of the talk. What I showed you is the development of this toolkit of low molecular weight humanized transcription regulators, we call synzifters. We use these to build clinically optimized gene circuits in primary T cells that can be controlled by orthogonal FDA-approved drugs. And we demonstrated more complex circuitry where you can actually enact multidimensional control over different payloads and thus potentially different aspects of T cell biology, proliferation, anti-tumor activity, et cetera. This is all kind of under the umbrella of what I called user control, just as a shout out to some work we did in collaboration that was really led by Cole Roybal's lab. We've also engineered, or his lab has engineered receptor systems, alternative receptors that can be expressed in T cells that drive gene regulatory programs. This is a new version of those of you familiar with the SYN-Notch receptors. What's nice about these new snipers is that they are entirely humanized, they're compact, and they're fully tunable. So we can even get these to work with our zinc finger transcription factors, the synzifters as well. It's nice to see that these have already now been licensed and are now in clinical trials. And some prelude to some newer work in the lab. We've been always looking to make these synzifters more active. They work pretty well in T cells, but you can imagine across other cellular contexts, we're going to really want robust activators. And so Bell, Sam, and Thea have undergone a really nice, nothing sort of on the order of what Mike Bassick and Lakra Bintu's lab has done, but something just more directed to look for small effector domains that when inserted into the synzifter platform can give us better transcription. And they have happened on a really fascinating, really small domain that seems to impact on elongation machinery that is giving us incredibly nice boosts in activity, where we're getting our synzifters now, we'll call them 2.0, that are achieving activity on the order of even surpassing these super potent viral activators like Viper. So we haven't published on this yet because we're studying this mechanistically, watching how, for example, can we stimulate elongation, and is that the mechanism by which we're really achieving these higher activities? But we're beginning to send these out because people are very interested in them. So if you're interested in 2.0 synzifter, you should reach out. That's good. All right. So I have about 10, 15 minutes. I'm going to give you the last bit of this, and it's going to be a shorter story. Is that okay? Does everyone still have energy? Okay. So this last bit, what I'm trying to sort of propose here is a more futuristic idea. And the idea is, can we use the reconstitution principles and building genetic circuits in cells to effectively teach cells, like let's call it yeast, which have no intrinsic therapeutic potential? Can we teach them to become, or can we train them to be basically powerful engines for generating therapeutically relevant molecules? All right. And to do that, we'd like to leverage what I think is the most powerful and innovative process that we know of, right, Tina? Evolution. Okay, so this work, which started about 3 or 4 years ago in the lab and is really coming to a nice place now, has been driven by a number of amazing graduate student postdocs. I'll talk mostly about Adam's work, but he's been supported by Arjun, Brandon, and Niket. And this is in collaboration with my good friend, Cheng Liu at UC Irvine. Okay, I'm gonna take us out of the nucleus for the last bit. Is that okay? We're gonna go to the membrane. And we're gonna talk about GPCRs. G-protein coupled receptors, of course, are these really important, physiologically important membrane proteins that act as diverse sensors of really, a lot of diverse signals, from hormones and peptides to light to odorants, et cetera. And they transmit that information into the cell to actuate responses. They're the largest class of membrane proteins in humans. We have, 800 or so. And over the years, skip through that, we've come to really appreciate the complexity of signal transduction by GPCRs. And its modulation. And I'm like condensing a vast, vast literature in biology in one slide here. But at a very basic level, GPCRs, recognize ligands, undergo conformational changes upon ligand binding, right? And then you get a wave of signaling. And so ligands can act as agonists that activate signaling, antagonists that suppress it, and even inverse agonists that do the opposite of agonism. And there's a whole set of G protein subtypes that act as transducers, and even other proteins like beta arrestins, effectors, et cetera. So there's a whole complex, signaling scheme here. And I will just sort of sum that all up and say, in a clause here, and say there's been really an explosion in our understanding of this biology. But I'll claim that our ability to generate synthetic, precise modulators that can capture, test, and perturb these different signaling dimensions on said GPCR, your favorite GPCR, is really limited. But of course, if we can readily make you a ligand or a synthetic modulator of a GPCR, I think it opens up a lot of really interesting possibilities for basic biology, for synthetic biology, but also for therapeutics, because as many of you know, GPCRs are one of the most prolific and famous targets of drugs. Okay, so what I'd like to convince you is that we can use yeast to solve this big problem, okay? Mighty budding yeast that gives us our beer, wine, and bread. And the way that we're envisioning doing this is the following. And we benefit from some really amazing things about yeast. The first is that yeast has actually been a model for studying human GPCR signaling for many years. And the reason for that is there's this great conservation in signaling pathways between the pathways that yeast use to mate and mammalian GPCR signaling, G protein signaling. And so what that means, because of that deep conservation, you can actually functionally express mammalian GPCRs in yeast. And if you do it right, they'll actually be functional. And in fact, you can couple them to G protein signaling in yeast. All right? This opens the door to doing really cool stuff, like building genetic circuits, what we do really well. For example, taking that signaling output and connecting it to, let's say, cell viability or some other selectable phenotype. And doing things like this. What we've devised is basically kind of like a 2-hybrid-like system, where we're co-expressing on the yeast membrane an anchored protein that can act as, let's say, a ligand or a modulator of that GPCR, okay? Such that if it activates or antagonizes the GPCR, we can select on that. So you can now put libraries of these said proteins, okay? Does this make sense? The other really neat aspect about yeast is, as many of you know, over the last few years, there have been really nice advances in developing continuous evolution, continuous directed evolution systems. Here's one example. This is developed by Arjun Ravikumar and Cheng Liu, called OrthoRep. You know, in a nutshell, this is a directed evolution system where you can encode a gene of interest on a plasmid in yeast, and that becomes hypermutated at now 1 million times faster than the mutation rate of, you know, the genome. And so you can basically, by just passaging yeast, hypermutate these genes. You're basically turning yeast into a search engine, right? And so you can, for example, encode this membrane-anchored ligand, for example, on OrthoRep, and then as yeast are just growing under selection, presumably select for activators, antagonists, et cetera, et cetera. Does this make sense? That's the vision. It's taken years to get this to work. But I'll just show you a little bit of data. This is actually just prototyping these selection circuits using actually the native yeast GPCR ST2, which controls mating. And what we're doing here is just co-expressing the native ligand of ST2 alpha factor and mutants of it. And what I'm showing you here is using signaling to drive a selectable and auxotrophic, you know, selection. And here, really nicely, you could see growing under selection, we can, you know, do positive selection for agonists and negative selection as well, okay? So this opens the possibilities for putting libraries and doing plate-based or even at some point liquid selection under and enriching for library members that can hit those GPCRs. What Adam has done is he's focused on one set of GPCRs for the moment as a pilot, and these are the muscarinic receptors. These are very interesting receptors for a number of reasons and a good challenge to our system. There's a subfamily of 5 receptors called M1, M2, M3, et cetera, M5. And they regulate functions of the nervous system and as well as a bunch of other things. What's interesting and challenging for them is that they have they're highly similar in sequence and structure. And this is like really foiled attempts to make modulators or drugs that can be subtype selective. Does that make sense? So many of the drugs that are actually known to work on these hit all 5 of these receptors. In addition, some of these receptors like M3 are just like they're expressed all through the body, right? And so there's actually, you know, pretty well-known antipsychotic small molecules that target dopamine receptors, and they have off-target activity on M3 in the pancreas, and this has like major side effects for these antipsychotic drugs, just as to give you an example of how difficult it is to get both tissue and subtype selectivity for these. So here's what Adam is doing. He's, it turns out that nature has somewhat solved this problem to some degree. Mamba snakes, as part of their toxin, express these mini proteins. We call them muscarinic toxins, MTs. They're really fascinating proteins, by the way. They look a little bit like antibodies, but they're mini. And they interact with muscarinic receptors. They kind of modulate antagonism, and this is part of their, you know, toxin, you know, function. And so what Adam is doing is using these as a privileged scaffold. We know they already interact with muscarinic receptors. Can we evolve them in a way, right, to direct their signaling activity? Can we make them into different modulators of muscarinic receptors? So here's like actually relatively new data that we're quite excited about. So what Adam did is encode a library that he generated of these mini toxins, muscarinic toxins, and put it into these selectable strains that I told you about. And here's kind of what some of these selections look like. So this is the M3 receptor. Okay, so let me just walk you through this a little bit. So there's a chemical agonist to M3 called carbachol. And so what I'm showing you here in this slide is if you don't display anything, right, you can drive activity and growth by adding different levels, increasing levels of carbachol. Does that make sense? And now what he's doing is he's selecting for variants that can act as what we call PAMs, not your PAMs, positive allosteric modulators, right? Ones that sensitize, right, to lower levels of carbachol. So he has a variant, he has a hit that seems to have really nice PAM activity on M3. He did the same experiment with the M1 receptor. And this we still don't understand. This is an amazing result. He seems to have found a hit that is acting like a pure agonist on M1. This defies my understanding because it's unclear how these toxins could even get into the orthosteric site. So they must be doing something very interesting here. So we'll see what happens. So what he's doing now is we're purifying these toxins, of course. We have developed a nice collaboration with a GPCR lab in biochemistry, where we can actually take and express receptors on mammalian cells. We can do really beautiful controlled signaling assays on these. And so he's got a couple of these purified, and we're beginning these studies now. So that's where we are here. I haven't shared with you kind of a whole set of other projects that are using similar circuitry to select for nanobodies that can interact with GPCRs. If you're interested, come talk to me. But I think we have now developed nanobodies against something like half a dozen GPCRs that have been unaddressed previously. So we're really excited about where we stand here. So here's the vision. Oh, whoa. Told you I'm excited. Here's the vision. We want to be able to just grow yeast cells under selection in high throughput and allow them to search space and give us potential therapeutic proteins, right? So with that, I'm just going to stop. I'll acknowledge funding over the years. I've already sort of acknowledged the individuals who are leading the projects. I have a wonderful group of collaborators. This is not comprehensive. Here are my conflicts. I want to thank you all for the invitation to be here, for attending the talk, and I'm happy to take questions if you have any. Thank you. Scott, yeah.
Hello. Great talk, Mo. I feel like in the first two arcs, you kind of presented two different solutions to the specificity problem, one based on cooperativity, which was really appealing, and the second one that was kind of like a computational negative selection in terms of, finding zinc fingers that had no cross-reactivity. And I was kind of curious which ones do you feel, or which one is, maybe the one is better for engineering and one is more evolutionary, or do they kind of work? You think they work in tandem in real genome?
Great question. Here's what I can tell you. The zinc finger solution. So the second part of the talk was making these 6-finger covalently linked zinc. They're two. This solution that's 2 by 2 by 2, right? And this has been like a common way of making, targeted zinc fingers against endogenous genes. Sangamo and et cetera have been doing this for years. And we followed that rubric. What Scott brings up a great point, right? So these are just designed in a positive way without any sort of other, solution. However, work by Marcus Noyes at NYU, who's developed like really great zinc finger engineering techniques, has, inspired by our work, introduced those arginine mutations that I was telling you about. And he's actually systematically explored these way more than us. I'm like embarrassed that he's done it. We haven't. But he's introduced some of these mutations that seem to lower binding energy. We think there's just excess binding energy in these designed proteins, right? And if you just back off a little bit by introducing those mutations, what he found in his paper, it was published in Nature Biotech last year, is you actually increase the specificity profile, okay? So that just one caveat to that is that people have started using that strategy, at least in one way, which is to at least just lower binding and rely on the flexibility of those what's called disrupted linkers between the two. So it looks a little bit, it's not quite the assembly that I showed you in the first talk, but then it begins to look a little bit like that. You and I talked a little bit. I want to implement a more cooperative system in mammalian cells. There's some technical difficulties of why that's been more challenging. Primarily, we're looking for good protein interactions that are quote unquote weaker, that are also designed, which is actually a difficult problem. But when we do, we will be implementing this. So I firmly believe that by using the principle of multivalency and cooperativity, I think is a winning strategy long-term to many of these genome targeting solutions. And I would love to see them, and we will over time, implement these types of things, not just in these kind of synthetic transcription factors, but in epigenetic editing and genome editing and even transcription factor systems where we can target endogenously. And I think the other thing to note, Scott, is I'm not sure if I'm supposed to talk about this, but as an SAB member of Chroma, which is developing epigenetic editing tools, they have been introducing the same mutations into their platforms and seeing also enhancements in specificity, which gives us a lot of confidence as well that these types of strategies, even the first idea, just like lowering affinity a little bit, is working. Does that answer your question? Yeah. All right. Yeah. Yes, in the back.
Hi, Mo. Great talk.
Thank you.
Going back to the first two arcs again, one of the things to consider about synthetic biology is like a lot of it has relied on looking into nature and then taking parts that do the things that we want and then piecing them together.
Yeah.
And it seems like that's how a lot of synthetic biologists, including yourself, have built their circuits.
Yeah.
But I just want to ask if we could, or if we should, but should, what is it? Ask whether or not if we could, not if we should question. We're in the advent of the alarming rate at which like deep learning models for protein structure prediction have been developing. If instead of looking towards nature, you can just computationally like design things for like transcription factors where they're modular, they need to like bind to certain parts, form complexes where you can almost go away from the traditional synthetic biology approach and then just kind of like design things that don't exist to do the things that you want them to do. So you can kind of avoid the search and what that would mean for the space.
Yeah. I think I like the trending to the sort of philosophical. That's nice. It's a good question, actually. I am equal opportunity. Let me just put it that way, right? We are inspired by nature. We draw from nature. You know, we have been drawing on nature with zinc fingers for many years, for example. I also firmly believe that computation, protein design, machine learning, all these techniques, right, are going to provide yet more sequence and fodder for engineering, right? But even just this problem that I was talking with Scott about here and answering this question, I don't know of any computationally designed protein-protein interaction in which you can dial in an affinity in the micromolar range and yet be specific and active. I don't even think David Baker has designed such a thing, right? And that's because PDZs have been sculpted through negative selection and positive selection over billions of years, et cetera, right? And so we should not forget that, right? And so it would be nice to train models on the diversity of PDZs and learn something there, right? For example, right? So the other thing is like, again, I think like, I'm not sure that computation is quite ready for us to sort of design a multivalent complex, right? That is, so I think all I'm trying to say is that I think we should be equal opportunity. We should look for ways in which we get for the design problem in mind, right? As a good starting point. We can use either nature or, as computation and as models, right, improve over time, use them when appropriate. Yeah. It's a very inarticulate way of answering your question, sorry. Yeah. Bill.
You set up your yeast selection for GPCRs.
Yeah.
And you mentioned this problem of the different orthologs, M1 through 5, and trying to achieve specificity. Are you able to set up some sort of positive, negative selection within a single yeast cell where you're looking at all of them and like reading out the activity you want?
Awesome question, Phil. In the other circuit that I didn't talk about, the hybrid circuit, we build a negative selection simultaneously in the same cell where we can select negatively against a background GPCR or closely related GPCR. And the results are striking. We can get like beautiful activity on the target GPCR and it just doesn't even touch, right? So we've done that already in these strains. Interestingly, for the muscarinics, that data I showed you there's no negative selection against the other subtype. And I haven't shown you this data, but those guys are exquisitely subtype selective, those toxins, at least in yeast. We get that for free somehow. It's a good problem for you to think about. Yeah, so somehow we got it for free with the muscarinics there, but in our other systems, we are doing these kind of negative selections, yeah. Mm-hmm.
Well, if there are any other questions, I guess let's thank Mo again for his talk,
you.
and thank you all for listening.
Thank you.